Hi, today I'm going to walk you through a basic example of doing differential gene expression with a library called DE-seq2. We're just going to go through the overall process to kind of lay out why we would do differential gene expression analysis, really what it tells us biologically. So for this tutorial and example, we'll be using an RNA-seq experiment that involved a handful of different samples collected from the cerebral cortex in mice. I've indicated the samples that we'll be comparing with our differential expression analysis here with different emojis. So our control samples are these three here at the bottom, indicated by the mice, and these are just healthy mice with no kind of intervention on them. These three samples that are labeled here as a 24-hour time point came from mice that were induced to have a stroke, and this is a 24-hour time point. So we'll be taking those samples and running through RNA-seq, and the typical output of an RNA-seq experiment after you've processed the data would be a count matrix like we're showing here. So in this count matrix, we have a column of gene symbols, and then in the subsequent columns are the other samples that were in our experiment. So here are our different 24-hour stroke samples, and then our three control samples here at the back. Inside of this count matrix, we have raw count values for each of the genes shown in the rows. So when doing differential gene expression, or really when starting my comparison to begin with, I may have a single gene of interest that I'm interested in looking at expression values. And if I have that, I would be able to create a relatively simple plot, say a bar plot or box plot, that shows the expression of that gene, say A2M, in my two different groups that I'm comparing here. So in this case, our control samples, which are these three mice, have very low expression of A2M. And then our three stroke samples here, indicated by the brain, have much higher expression of A2M. And if I'm just looking at a single gene like that, I could, for example, run a two-sample t-test to compare just between my 24-hour or my no-stroke group here, what that p-value looks like. And I can see here that if we compare these two groups, this is a very statistically significant difference in the expression of the A2M gene. However, this is only a comparison for one gene at a time. But we know that in our experiment, we might have 30,000 or more genes that are being quantified by a whole transcriptome RNA-seq experiment here. And that's really where differential expression comes into play. We want to be able to do this kind of a comparison, but for all of the genes that were measured in our assay, and then use the correct statistics to adjust for the fact that we're doing this kind of comparison, but 30,000 plus times. So for a differential gene expression analysis, I'll create a new analysis, choose differential expression analysis, and now you could write your own scripts for performing differential expression or use a no-code platform like Pluto to do this analysis. I'll create a new analysis for us here, and referring back to my sample data sheet, I know that time point is what I'll be comparing my samples by. So I'll select time point, and then set up a comparison comparing the three 24-hour samples compared to our three samples that did not have any stroke. And click Run to run this bioinformatics pipeline. The typical output for differential expression is a volcano plot. This is a pretty common output way of visualizing this data. On the x-axis here, you'll see the log2 fold change of genes, which are represented in this case by different data points on a scatter plot. And the log2 fold change indicates the magnitude and direction of the change in expression. So here we can see A2M, the gene that I was using before, is very upregulated, meaning it's on the right side of our x-axis here. And then it's also very statistically significant. So on the y-axis, we're indicating statistical significance, and that's a negative log 10 transformed adjusted p-value, which means that values on the higher end of this y-axis are more statistically significant, or a lower p-value. And so you can see that here. If I make this p-value more significant, you can see the corresponding negative log 10 adjusted p-value here. And I can also show that as a threshold on the horizontal line here. So the genes that have been increasing in our 24-hour stroke condition now, we will highlight in red just to indicate that change. So now we can see that as opposed to looking at a single gene, A2M at a time, we have a visualization that indicates to us how all 30,000 plus genes in our experiment were changing across these two different conditions. This can also be represented in a tabular view, which is typically called a differentially expressed gene table. So here you can see A2M is at the top of our list based on assorted adjusted p-value values. But we have additional genes here shown in this list, and they can be ranked by their fold change or adjusted p-values. It's always important to take into account the methods that you're using when doing a bioinformatics analysis like this. So Pluto has these handy method sections that show you the p-value threshold that we were applying in our visualization, the exact comparison that we were doing in this case, so 24-hour stroke versus our no stroke control, and then taking into account the algorithm that you were using. So for this analysis, we were using a library behind the scenes called DE-seq2 to do the statistical analysis and calculation of the p-values shown in this volcano plot. That's a basic introduction to differential gene expression analysis and why you would create something like a volcano plot over looking at one gene at a time in a bar plot in your RNA-seq data. Thanks so much for watching, and we hope to see you soon in Pluto.